In the first few weeks of life, far before anyone even knows these systems exist, the embryo is busy constructing the future capacity for both filtering waste and, facilitating reproduction.
And there's such different functions.
Wildly different. Yet, they somehow share a common, cramped construction site, often using the exact same starter parts.
It truly is the ultimate lesson in biological reuse and repurposing. I mean, what starts as temporary plumbing for a rudimentary waste filter becomes the foundational ductwork for the entire reproductive tract.
That's incredible.
What's fascinating here is how fundamentally interconnected these systems are. Initially, they're sharing not just physical space, but the same origins in the mesoderm, and even the same temporary ducts.
Right.
And that, it highlights a certain developmental fragility where a single misstep in timing can affect two entirely separate functions decades later down the line.
Exactly. And that's our deep dive today. We're getting into the source material covering the detailed embryogenesis and development of this very urogenital system. It's a journey that traces structures from these initial cellular migrations all the way to fully formed neonatal organs. And it highlights some incredible transformations and, frankly, some anatomical connections that seem almost too clever to be random.
They really do. Our source material, spanning detailed chapters and illustrations, maps this entire intricate development, from the initial formation of the column wall through the succession of 3 separate temporary kidneys, all the way to the definitive hormonal differentiation of sex and the eventual robust function of the metanephros, which is the permanent kidney, at birth.
And our mission here is critical.
It is. We are unpacking the chronological stages, the key structures, and the surprising clinical implications that arise when these processes deviate even slightly from the script.
Okay, let's unpack this. This is dense molecular history carved into our earliest stages of life. We are talking about 3 successive kidneys, pronephros, mesonephros, metanephros, that appear and then just disappear.
Right.
And a dual reproductive blueprint existing simultaneously in what is called the indifferent stage. It's truly a story of temporary scaffolding, permanent construction, and then this final irreversible hormonal decision.
And we will analyze not just what develops, but the precise timing and the underlying genetic and protein signaling that dictates why one developmental path is chosen over another.
Right.
And we also have to acknowledge, right up front, that our detailed knowledge on the development of the meso and metanephros in living human embryos is frustratingly limited.
That's a huge caveat.
It is. Much of what we know relies instead on advanced experimental systems, like stem cell-derived kidney organoids and careful studies of comparative embryology. And this just reminds us that we are constantly relying on these extrapolated models to understand these absolutely fundamental human processes.
So let's start at the very foundation of this whole massive endeavor, the posterior abdominal region. We're talking around day 29 post-fertilization, which is designated as stage 11 in embryonic development. To set the scene, what exactly is defining the boundaries of where this construction project begins?
Okay, so the starting point is the wall of the intraembryonic column. Our source material describes this wall as being composed of 2 crucial cell populations. The first is a mesothelial population, which forms the outer lining, sort of the future parietal layer.
Okay.
And the second is a population of somatic cells just underneath that. The mesothelial cells are absolutely vital because they line this body cavity, and their movement defines exactly where the new organs are going to appear.
And their movement is dictated by a specific migratory route. I see a description in the sources of these cells undergoing ingression through the primitive streak. This isn't just, a casual stroll. It's a critical migration event.
That's exactly correct. The mesothelial population undergoes this deep specific ingression through the primitive streak, and the precision of this process determines their final fate and their final location.
Right.
What's fascinating is the specific cellular choreography involved. These urogenital-bound cells, they always trail the cells that are forming the lateral plate of the mesoderm.
Oh, interesting.
So you can think of the lateral plate cells as the advance guard, laying down the road, and these mesothelial cells are following immediately behind.
Why is that trailing timing so crucial? What happens if they get there too early?
Well, that precise timing ensures the cells settle in exactly the right place, specifically forming what we call the intermediate mesoderm in the posterior abdominal region. If these cells were to arrive too early or, too late, the entire nephrogenic cord, which is the spine of the developing system, would be misplaced relative to the surrounding structures.
Then that would be catastrophic.
It would guarantee systemic failure. It's a perfect illustration of how developmental processes stack upon each other. The success of step 1 dictates the possibility of step 2.
So the intermediate mesoderm is the primary raw material here. Once it settles, what form does it take? Does it just stay as a sheet-like structure?
No, it organizes immediately. The intermediate mesoderm, which is crucial for all subsequent urogenital development, it forms a distinctive longitudinal column that runs down the embryo. And this column is very appropriately named the nephrogenic cord.
Okay.
This cord is established right near the anatomical junction between the splenopleuric mesenchyme, which is adjacent to the endoderm, the inner digestive layer, and the mesothelium, which is adjacent to the ectoderm, the outer layer.
Yeah.
So it is precisely sandwiched between the three major germ layers in the posterior region, and that highlights its central role in mediating these external and internal structures.
That location, the urogenital ridge, it really is the developmental hub. What happens in that space defines the course for the next 3 months of growth. And the definitive calum wall is also forming simultaneously from this activity, right?
Indeed. The final definitive calum wall isn't just that initial mesothelium, it's the result of the proliferation of that overlying calum epithelium combined with the adjacent intermediate mesoderm and the splanchnic mesenchyme.
So it's a team effort.
It's a huge team effort, and this complex, integrated interaction is what generates the urogenital ridge, a large bilateral structure that defines the construction zone for the kidneys, the adrenal glands, the gonads, and all the associated reproductive ducts.
This shared mesodermal origin is probably the most profound theme of this entire deep dive. It's why all these systems are so inextricably linked.
Absolutely. The kidneys, the adrenal glands, the gonads, and the reproductive ducts all arise from this single foundation. If you ever wondered why, for instance, kidney anomalies often coincide with reproductive tract anomalies, this shared mesodermal heritage is the fundamental reason. They literally grow up together in the same neighborhood.
That sets the stage perfectly. Now we need to look at the overall developmental strategy for the excretory system, because the body does not build the final permanent kidney, the metanephros, first. It uses these temporary successive models, almost like evolutionary prototypes.
That's a great way to put it. The renal excretory system, as detailed in our sources, develops from 3 successive organs, each derived chronologically from that intermediate mesoderm. The pronephros, then the mesonephros, and finally the metanephros.
So it's a 3-stage evolution, and it kind of mimics structures that we see across various vertebrate lineages.
Precisely. And the importance of this shared heritage is that while these systems succeed each other in time, they also succeed each other in space, moving from cranial, so the head end, down to caudal, the tail end. The subsequent systems always develop just caudal to the preceding one.
A clear progression.
A highly reliable chronology observed in all human embryos. It is the body essentially practicing the necessary steps before building the permanent version, making sure the correct developmental pathways are engaged.
And this brings us back to that crucial caveat you mentioned right at the start about the clinical knowledge gaps. If this chronology is so reliable, why is our understanding of the meso and metanephros still incomplete?
It comes down to observation limits. The detailed development of the meso and metanephros occurs deep within the embryo during a period, weeks 5 through 9, that is just incredibly difficult to access or study ethically in living human embryos. So we have to rely heavily on these advanced experimental systems like stem cell-derived kidney organoids, which allow us to mimic the tissue induction and growth in a petri dish.
And animal models.
And we also rely on careful studies of comparative embryology, looking at species where development might occur more slowly or visibly.
But that reliance on models creates potential issues, I would imagine.
It does. We have to acknowledge that differences, what are known as heterochronic differences, exist between species. And that means the timing or the exact scale of certain events might be unique in humans compared to, say, a mouse model. This makes interpreting the fine-grained molecular signals challenging, constantly requiring us to factor in potential human-specific nuances when analyzing the results from these extrapolated models.
Okay, let's dive into the structures themselves, starting with the earliest and most transient one, the pronephros. In lower vertebrates, this structure is functional, sometimes permanently, but what's its status in human development?
Well, the pronephros is effectively an evolutionary echo. In lower vertebrates, the intermediate mesoderm organizes into these discrete serial segmented masses called nephratomes, and these nephratomes develop a cavity, the nephrocole, a duct, the nephrocole duct, and often a connection to the coelom via a peritoneal funnel, known as the nephrostome. That's a robust functional unit for simple waste processing in, say, a larval fish.
And in us, how rudimentary are we talking?
In human embryos, the proniferous is only represented by these transient clusters of cells located in the most cranial portion of the urogenital ridgeway up in the cervical region. Crucially, the sources emphasize it is considered rudimentary in mammals.
So it's there and then it's gone.
It may form the duct or some vestigial tubules, but it shows up briefly in the fourth week, regresses almost immediately, and it never persists or becomes functional in the robust way it does in lower forms.
So it checks the box evolutionarily. But the real developmental energy goes into the second set of kidneys, the mesonephros, which establishes the primary excretory duct system. Tell us about the timing and physical location of this second stage.
The mesonephros begins its development from the intermediate mesoderm, just caudal, to the regressing pronephros. Its emergence is timed around stage 11, roughly day 30-31 post-fertilization, which correlates with the embryo having about 5 to 8 somates, and it runs from the upper thoracic down to the upper lumbar regions.
This structure seems to be responsible for establishing the primary plumbing system for the future, which is the mesonephric duct. How does that crucial collector duct form?
The primary excretory duct, which we'll later call the mesonephric or Wolffian duct, it starts its life as a solid cord of cells that develops in the nephrogenic cord. This cord then undergoes canalization to form a true hollow duct. And this duct is actively growing caudally and eventually establishes a connection, opening into the cloaca, the common terminus for the hindgut and the urinary tract, by stage 12. This duct runs the length of the mesonephros, acting as its main collector.
And what about the actual filtering units within the mesonephros? Are they also derived from that intermediate mesoderm, and how quickly do they organize?
They absolutely are. They arise from the surrounding nephrogenic cord tissue. These are the mesonephric tubules. They arise segmentally, typically forming one pair per somite, and our sources estimate that up to 40 such structures can form transiently.
Wow, 40.
And each tubule opens directly into that primary excretory duct, and importantly, develops an internal glomerulus, a renal corpuscle, which provides the necessary filtration surface.
So this is a fully formed, albeit temporary, filtration system. Is it capable of managing the embryo's waste?
It is functional, but only for a limited time, specifically between weeks 5 and 8 of development. It serves a vital filtering role for the developing embryo before the placenta takes over most of the excretory duties.
And then it's dismantled.
It is. Once the metanephros, the permanent kidney, is ready to take over, the mesonephros rapidly regresses. The regression rate is striking. Studies indicate that individual embryos can lose between 30 and 49% of their mesonephric tubules in a short 4-week period. It's a very fast demolition job.
But that brings us back to the theme of biological reuse, doesn't it?
Yeah.
Even as this structure is being dismantled, it leaves behind some incredibly important legacy tissue.
That is the ultimate act of biological recycling. The majority of the tubules are lost, but crucial parts of the mesonephros persist in males to form the entirety of the internal male reproductive plumbing.
The whole system.
The whole thing. This includes the efferent ductules, which connect the testes, the epididymis, the ductus deferens, and the seminal vesicle. In females, conversely, these ducts largely degenerate due to the lack of specific hormonal stimulus, leaving behind only minor non-functional vestiges.
So the duct that carried temporary urine in the male embryo becomes the pipeline for sperm in the adult.
That's it. It's astonishing.
That is astonishing. Now we move to the star of the show, the final permanent kidney, the metanephros. Where does this structure take root, and what sets it apart from the two earlier temporary systems?
The metanephros develops far caudal to the regressing mesonephros, forming in the sacral and lumbar segments of the intermediate mesoderm. What sets it apart is its unique dual embryonic origin. It requires two completely separate components to induce, communicate, and form the final complex organ structure.
That dual origin is essential for you to grasp. So what are the two components, and what part of the final kidney does each component form?
Okay, so the first component is the ureteric bud. This is an evagination or an outgrowth that literally sprouts from the mesonephric duct near where the duct enters the cloaca. This bud is the precursor for the entire collecting system. All the plumbing, the ureter, the renal pelvis, the major and minor calyces, and all the collecting ducts that funnel the urine. The second component is the metanephric mesenchyme, which is derived from the intermediate mesoderm located further down in the sacral region. This mesenchyme is the tissue induced by the bud to form the actual filtering units, the nephrons.
The business end of the kidney.
Exactly. Bowman's capsule, the proximal and distal tubules, and the loop of Henle.
So we have two independent contractors, the bud and the mesenchyme, who have to communicate perfectly. This sounds like a biological conversation, a reciprocal induction where one part talks to the other to ensure development proceeds correctly.
That is precisely the mechanism. Our sources detail this intricate two-way communication extensively. The ureteric bud grows into the metanephric mesenchyme, acting as the initial messenger. The mesenchyme then stimulates the bud, telling it to branch and divide rapidly, a critical command. In return, the bud releases factors that induce the mesenchyme to condense, epithelialize, and transform into the filtration units.
And if that conversation breaks down,
If either party fails to send or receive the correct signal, the permanent kidney will be dysplastic or fail to form altogether. It's that critical.
let's focus on the plumbing first, the branching of the ureteric bud. How does this single outgrowth transform into the complex internal architecture of the kidney, the calyces, and collecting ducts?
It's this highly controlled and sequential process of branching, almost like a fractal tree. The first 6 sequential branching divisions are what form the wider funnels of the kidney, the major and minor calyces of the renal pelvis. These are the large collection points.
Okay.
Following that, subsequent smaller divisions, ranging from the 7th up to the 14th branching event, are responsible for generating the final population of hundreds of thousands of collecting ducts.
And as that branching tree is forming, the adjacent mesenchyme is condensing and forming the nephrons themselves. Walk us through how that mesenchymal cap becomes a highly structured, S-shaped tubule.
So the metanephric mesenchyme cap, which sits directly over the tip of these collecting duct branches, transforms first into what are called metanephric vesicles. These vesicles then elongate and transform into the classic, visually recognizable S-shaped tubules.
Right.
And it's the differentiation of these S-shaped tubules that leads to the fully formed nephron, the proximal tubule, the loop of Henle, the distal convoluted tubule, which finally connects perfectly to the collecting ducts originating from the ureteric bud. It is a continuous assembly line, meaning that the most recently formed and therefore least mature nephrons are always located near the outer cortex of the kidney.
The functional core of the nephron is the glomerular filter, where blood filtration actually happens. This structure is famously precise, blocking large proteins but allowing waste through. What molecular complexity is required to build this highly selective filter from a simple S-shaped tubule?
This is where the molecular signaling becomes incredibly fine-tuned. The S-shaped tubule must differentiate into several cell types simultaneously. The part that encapsulates the capillary loop becomes Bowman's capsule. And crucially, the inner epithelial cells of the S-shaped loop differentiate into the visceral epithelial cells of Bowman's capsule, these specialized podocytes.
Ah, the podocytes and pedicles. This is where a good analogy really helps.
Absolutely. Think of the podocytes as having long, thin fingers that interlace with the fingers of adjacent podocytes. They don't fuse fully. They leave these tiny, precise gaps. These are the filtration slit diaphragms. And this interlaced structure acts as an ultra-selective sieve, allowing fluid through but trapping large plasma proteins.
What are the key molecular players holding that sieve together?
Well, that structural assembly requires precise molecular signaling. We see evidence of complex signaling involving factors like platelet-derived growth factor B or PDGFB interacting with its receptor, PDGFR-beta. This interaction is key in generating the glomerular basement membrane and promoting the development of the mesangial cells, which support the capillaries.
And the slits themselves.
Right. Specialized proteins are required to form those filtration slit diaphragms, primarily nephrin and pitocin. If you have an impairment of these molecules, you disrupt that sieve function, and the result is severe protein leakage, leading to congenital nephrotic syndromes.
That provides crucial detail in the formation of a single nephron. But the sources also emphasize the importance of maintaining the cell population that keeps generating new nephrons throughout gestation. It's like a renewable factory floor.
Yes, the constant renewal is fundamental. The source material specifically highlights the critical role of Wnt signaling in maintaining that progenitor population, the self-renewing mesenchymal cap cells.
So what is the keep going signal?
Yeah. You can think of it as the continuous keep dividing signal, ensuring the mesenchymal cap doesn't run out of raw cells. If that Wnt signal drops prematurely, it's like shutting off the assembly line. The consequence is immediate. The supply of progenitor cells is depleted, which can lead to kidney hypoplasia, an underdeveloped kidney, or dysplasia.
And while the nephrons are being constructed, the essential process of vascularization must be happening, bringing in the blood that the nephrons are meant to filter.
The vascularization is intricately linked and managed by the angiogenic mesenchyme that migrates in. Vascular endothelial growth factor, or VEGF, is a key molecular signal here, promoting the formation of the capillaries. Capillary loops grow into the S-shaped tubules and integrate perfectly with the developing glomerular structures. The mesenchyme surrounding the nascent nephrons plays a key regulatory role in establishing and maintaining proper blood flow to the final renal vessels.
So how long does this incredible high-speed assembly line run? When does the kidney finish production?
Nephron formation is generally completed by week 34 of gestation, though maturation continues. This is a crucial timeline because any infant born before week 34 has a congenital deficit in nephron number. They are born with fewer filtering units than a full-term infant.
A lifelong
A
deficit.
lifelong deficit. Our sources indicate that at full term, the kidney weighs about 23 grams and is still somewhat lobulated. The physical growth during the third trimester is phenomenal. The length of the renal corpuscle, including the loop of Henle, increases from a tiny 83 micrometers up to 849 micrometers. And this physical growth and functional maturation continues rapidly in the postnatal period, especially in the first 2 years of life.
Okay, so now we have the permanent kidney structure, the metanephris, constructed down on the pelvis. But we need to connect it to the rest of the body, which requires a major geographical shift, the kidney ascent.
Yes, this relocation is really counterintuitive. The metanephris initially forms quite low down in the sacral region, near the common iliac vessels.
Right, so how does the kidney manage this seemingly impossible move up to the adult position in the flank?
The ascent process is not about the kidney actively climbing, but rather it's the result of the differential growth of the lumbar and sacral regions of the body. As the rest of the embryo elongates and the lumbar curvature changes, the kidneys appear to ascend to their adult position.
And they rotate too.
As they move, they also rotate medially, so that the hilum, the entry point for vessels, and the ureter changes from facing ventrally to facing anteromedially.
That relocation requires a complete sequential swap of blood supply, doesn't it? It must be a series of dangerous arterial handoffs from the aorta.
That is exactly right. As the kidneys move up, they receive new, successively higher level arterial supply from the aorta. Concurrently, the lower original vessels, which often stemmed from the iliac arteries, typically regress. It's a beautifully choreographed series of connections and disconnections.
But sometimes it doesn't go perfectly.
Right. Failure of this full regression process can lead to the persistence of those lower vessels. The source notes that supernumerary arteries, where some of the transient lower vessels persist alongside the definitive renal arteries, are actually a common variation seen in angiography.
Let's track the plumbing now, the ureter. This is the duct connecting the newly ascended kidney to the lower system. What's its origin, and how does it successfully integrate with the developing bladder?
The ureter is derived entirely from that ureteric bud. Initially, the ureter connects to the mesonephric duct right before it enters the cloaca via a segment called the common duct. As the embryo grows, this common duct segment shortens dramatically, and this shortening is crucial because it allows the ureter and the mesonephric duct to separate completely.
And that separation ensures they don't open into the same place permanently, which is vital for the gender-specific plumbing later on. What happens next to the ureter?
The ureter then migrates and opens directly into the developing cloacal region, which is the future bladder. Simultaneously, the mesonephric duct shifts caudally and medially, ensuring that the urinary and reproductive outflows begin their necessary separation.
This brings us to the common receiving tank for the urinary and reproductive tracts before that separation. The cloaca. How does the cloaca, that shared terminal space, transform into the bladder and the other ventral structures?
The cloaca is the terminal end of both the hindgut and the allantois, a crucial shared space. This common area is partitioned by the urorectal septum, which is a mesenchymal wedge that grows downward and fuses with the cloacal membrane.
Like a wall growing down the middle.
Exactly. And this septum eventually divides the cloaca into two distinct parts: the dorsal structure, which forms the rectum and anal canal, and the ventral structure, which forms the urogenital sinus.
So the urogenital sinus is the primary ventral structure for the entire system we're tracing. What are its major derivations?
Well, the urogenital sinus is the source of multiple structures based on its three distinct regions. Its cranial portion becomes the definitive urinary bladder, the large main reservoir. The intermediate pelvic portion forms the prostatic urethra and membranous urethra in males, and the entire female urethra. And finally, the caudal phallic portion contributes to the external genitalia.
And the bladder lining itself.
Critically, the bladder epithelium itself is derived from the endoderm of the urogenital sinus.
Now, for one of the most interesting parts of the bladder anatomy, the trachone. that smooth triangular area at the base of the bladder defined by the ureteric openings. The sources mention its origin is unique.
Its formation involves a remarkable process where the lower ends of the mesonephric ducts and the ureters are literally absorbed into the developing bladder wall tissue. Yes. As this happens, the ureters migrate laterally and cranially, establishing their separate openings. The tissue that forms the trigone, the area between the ureteric openings and the bladder neck, is the mesoderm-derived tissue that originally surrounded the common duct. This mesoderm-derived tissue persists and expands, forming the trigone.
Wait, that means the rest of the bladder lining is endodermal, but the trigone is mesoderm-derived from the old duct systems. That's a stark difference in cellular heritage within a single organ.
Precisely. The trigone is mesoderm-derived, a distinction with profound clinical relevance. The source material emphasizes this, noting that the differing epithelial characteristics of the trigone reflect this change in origin. But the most critical clinical application relates to how the ureter passes through the trigone.
How does that precise integration link to pathology?
The precise geometry of the ureter as it passes through the muscular wall of the bladder is what determines valve function, preventing reflux. Improper fusion or separation of the ducts during absorption can lead to a shorter intramural segment of the ureter or an abnormal angle of insertion.
And that breaks the valve.
It compromises the natural valve mechanism, leading to conditions like vesicoureteral reflux, VUR, where urine flows backward from the bladder toward the kidney, potentially causing kidney damage and recurrent infections. The structural difference in the trigone's mesodermal tissue compared to the muscular endodermal wall is central to this mechanism.
Let's look at the final picture. Kidney function and growth at birth. We know the fetus is constantly producing urine into the amniotic fluid. What are the key function metrics for a full-term kidney?
Fetal kidneys function early, contributing significantly to amniotic fluid volume, which is vital for lung development. By full term, around 40 wints, they weigh approximately 23 grams. However, the period immediately following birth is characterized by rapid maturation because the neonate needs to transition from relying on the placenta to full independent renal function.
This is where the glomerular filtration rate, or GFR, comes in, the gold standard metric for kidney function. Can we quantify the functional deficit and the subsequent rapid increase?
We can. GFR is measured in milliliters per minute per 1.73 meters squared of body surface area. At 32 weeks of gestation, the GFR is remarkably low, around 1.73. By term, that increases sharply to about 12.3.
Still very low.
Extremely low compared to adult values, which typically range between 100 and 130. This acceleration of GFR continues throughout infancy, but that initial low rate highlights the kidney's immaturity.
So the newborn kidney is functionally fragile compared to an adult's. What does that functionally fragile status mean for a neonate in a practical sense? Why should we care that the GFR is only 12.3?
It means the newborn renal system is extremely sensitive to stress and has very little reserve. The low GFR and the kidney's limited ability to concentrate urine mean they are prone to dehydration and electrolyte imbalances, and they also clear drugs much more slowly.
So it's a clinical vulnerability.
A huge one. This is why newborn renal function is known to be particularly sensitive to stress factors like hypoxia, asphyxia, and prematurity. For instance, premature infants often require very careful fluid and electrolyte management due to their delayed maturation.
And the source material mentions one specific common anomaly seen prenatally that often resolves itself.
Yes. A common clinical observation is temporary moderate renal tract dilation, known as hydronephrosis, which is seen in 1 to 2% of prenatal ultrasounds. In many of these cases, it resolves spontaneously without intervention, simply because the system is still maturing and the exact relationship between the collecting system and the ureter may still be adjusting. This highlights the fluidity of development, even late in gestation.
We mentioned earlier that the urogenital ridge gives rise to the kidneys, gonads, and the adrenal glands. Let's now look at the adrenal glands, which sit right on top of the kidneys. The suprarenal, or adrenal gland, is another fantastic example of dual embryonic origin, much like the metanephris, but with entirely different cellular sources.
It is truly a composite organ, born from an incredible collaboration. The outer cortex is derived from the mesoderm, specifically from the coalomic epithelium adjacent to the developing mesonephris.
Okay.
The inner medulla, however, originates entirely from neural crest cells, which are ectodermal in origin.
So two completely different germ layers coming together. When does the cortex, the mesodermal component, begin to form? Does it happen early?
Cortex development begins relatively early, around stage 14, approximately 36 days. Cells from the coelomic epithelium start migrating and proliferating into the underlying mesenchyme near the mesonephros. This initial massive migration forms the fetal cortex. Later, a secondary wave of cells migrates in to form the permanent adult cortex, which is encapsulated by a fibrous layer.
And the medulla, the neural crest component. Those cells are famous for their ability to travel long distances in the embryo.
That's their defining feature. The medulla begins to develop slightly later, around stage 20. Neural crest cells, specifically sympathetic neuroblasts, migrate all the way from the neural tube region to the medial surface of the developing cortex.
And once they're there,
Once they are successfully encapsulated by the cortex, these neuroblasts transform into chromaffin cells. These are the functional cells responsible for producing and releasing catecholamines adrenaline and noradrenaline, which are critical for the sympathetic stress response.
one of the most striking anatomical facts about the fetal adrenal gland is its size. It's disproportionately huge. Why does the fetus need such an enormous adrenal gland?
It is indeed massive. The fetal adrenal gland can be up to 10 to 20 times larger than the adult gland relative to body weight, a fact that always surprises students. And this is entirely due to the huge size of the fetal cortex, which makes up about 80% of the gland volume.
And the reason is hormonal.
The reason is hormonal. The fetal cortex produces massive amounts of dehydroepiandrosterone sulfate, or DHEAS.
Why is that specific steroid important?
DHEAS is an essential precursor steroid. The placenta cannot synthesize estrogen on its own. It requires this precursor, DHEAS, from the fetal adrenal glands. Thus, the massive size of the fetal cortex is dedicated to providing the necessary substrate for placental estrogen synthesis, which is crucial for maintaining the pregnancy and maturing the fetus.
That immediately explains the dramatic structural change required postnatally.
Precisely. Since its purpose is fulfilled at birth, the fetal cortex is temporary and rapidly involutes postnatally, regressing significantly about 50% of its volume within the first year of life. As this involution occurs, the permanent adult cortex slowly develops from the subcapsular region, eventually differentiating into the three characteristic adult zones: the zona glomerulosa, the zona fasciculata, and the zona reticularis.
This entire complex interplay of hormones and development leads us directly to a key clinical relevance: congenital adrenal hyperplasia, or CAH.
CAH is a crucial example of how even minor enzyme deficiencies can derail the entire system, linking developmental biology directly to a life-threatening clinical scenario. It is one of the more common congenital disorders, with an incidence ranging from 1 in 5,000 to 1 in 15,000 births, depending on the population.
What is the underlying genetic and enzymatic cause of CAH?
CAH is most frequently caused by mutations in the CYP21 gene, which results in 21-hydroxylase deficiency. This enzyme is essential for synthesizing two critical hormones: cortisol, the primary stress hormone, and aldosterone, which manages salt and water balance.
So when that enzyme is deficient,
The body cannot produce enough of these critical hormones.
and if you can't run the main chemical pathways to make cortisol or aldosterone, where do the precursor hormones go?
They accumulate and are immediately shunted down the only available pathway that is not blocked, which is the pathway that produces androgens, or male hormones. So this results in a clinical scenario of deficient cortisol and aldosterone production, but a dangerous accumulation of androgen precursors. This accumulation is what causes the significant developmental consequences prenatally.
And those consequences are different depending on the genetic sex of the fetus.
Absolutely. In individuals who are genetically XX, so female, the massive early surge of androgens causes virilization of the external genitalia, meaning the external structures appear more masculine. The severity can range widely. Furthermore, in all affected newborns, regardless of genetic sex, the lack of aldosterone production can lead to a severe, life-threatening salt-wasting crisis in the first few weeks after birth.
That is a life or death emergency caused by a failure in an enzyme.
It is. The lack of aldosterone prevents the kidneys from retaining sodium, leading to severe dehydration, dangerously low blood pressure, and electrolyte imbalance. This condition illustrates the profound reliance of anatomical development, and indeed immediate survival, on the precise balance of fetal adrenal steroid production, all orchestrated by those mesoderm and neural crest cells.
We now shift focus fully to the reproductive system. Before any sexual differentiation occurs, the reproductive tract goes through a period of structural neutrality, the indifferent stage. Everything needed for both male and female development is laid out simultaneously.
This stage is characterized by the coexistence of rudimentary male and female structures. Everything begins within the urogenital ridge, which is situated medial to the mesonephros. This ridge, composed of coalomic epithelium and underlying mesenchyme, forms the gonadal ridge.
But the true precursors of the reproductive system, the gametes themselves, aren't native to that ridge, are they? We have to track the incredible journey of the primordial germ cells, or PGCs.
The PGCs are the true reproductive precursors, and their journey is truly one of the most remarkable cellular odysseys in the body. They form extremely early, around day 20 post-fertilization, originating far away in the wall of the yolk sac, outside the body axis of the embryo.
Why such a circuitous route? Why start so far away?
The exact biological reason is still debated, but one hypothesis suggests that the germline must be established very early and protected from the powerful inducing signals that are transforming all the somatic cells of the embryo.
So they're kept separate.
They are, and then they must actively migrate using amoeboid movement through the hindgut and along the dorsal mesentery, all the way to the genital ridges. They arrive around stage 11, where they appear as large, distinct cells that are positive for alkaline phosphatase, which helps us identify them.
Once they settle, what is the vital function of these PGCs? They are the initiators, aren't they?
They are the foundation of the gametes, and they are the essential inducers. They proliferate rapidly, and, crucially, they interact closely with the somatic cells of the surrounding mesenchyme. This interaction is the induction signal needed for the mesenchyme to condense and form the sex cords, the internal scaffolding of the future gonad.
So if they don't arrive,
If the PGCs fail to migrate or thrive, the resulting gonad will be underdeveloped or fail to form altogether, leading to what we call gonadal dysgenesis.
let's turn to the plumbing of the indifferent stage. Before the hormonal switch flips, the embryo has a full dual duct system, setting up the entire choice architecture.
Exactly. Around week 6, the embryo simultaneously develops 2 complete sets of parallel ducts running down the trunk. The first set, which we already know, are the mesonephric ducts, or Wolffian ducts. These are the pre-existing excretory ducts from the temporary mesonephros, and they are situated laterally.
And the second, new set of ducts.
Those are the paramesonephric ducts, or Müllerian ducts. They begin as invaginations or inpocketings of the coalomic epithelium lateral to the cranial end of the mesonephric ridge. They grow caudally, running parallel to the Wolffian ducts.
And they cross over.
As they descend, they cross the mesonephric ducts ventrally, fuse at the midline in the pelvis, and terminate on the urogenital sinus. So both duct systems are fully present, running their separate courses, awaiting the genetic signal.
Which brings us to the molecular mechanism that dictates whether those indifferent structures follow the male or female path. This decision is irreversible and dictates the fate of all that shared mesodermal plumbing.
It does. And this is all down to the master switch, as the sources call it, the SRY gene.
The SRY gene, or sex-determining region Y, located on the Y chromosome, is a master switch for testicular development. Its expression is what initiates the entire cascade for male development, triggering the gonadal cells to differentiate. And SRY expression begins around week 7 of gestation.
So, SRY is expressed, and the immediate cellular consequence in the gonadal ridge is that the supporting cells within that ridge differentiate into Sertoli cells.
And they take over.
The Sertoli cells immediately take command of the developing gonad, orchestrating the subsequent 2 critical hormonal events necessary for male development.
What are those 2 absolutely crucial hormones, and what is the specific job of each?
The first and most dramatic hormone is anti-Müllerian hormone, AMH, also known as Müllerian inhibiting substance, or MIS.
Right.
Produced by the Sertoli cells starting around week 8, AMH is the chemical bomb responsible for the rapid, almost complete destruction and regression of the paramesonephric or Müllerian duct system.
So it's actively getting rid of the female plumbing.
Exactly. This ensures a future male is not saddled with a uterus or fallopian tubes.
So AMH actively eliminates the female duct system. What is the second hormone, and what is its role?
The second hormone is testosterone, produced by the Leydig cells, which differentiate after the Sertoli cells, also starting around week 8. Leydig cells are initially stimulated by maternal HCG and later by fetal LH-FSH. Testosterone's primary role is active maintenance and promotion of the existing mesonephric or Wolffian ducts. It rescues them from their default fate of regression.
So in the male pathway, the duct fate is Müllerian ducts are actively destroyed by AMH, and Wolffian ducts are actively maintained by testosterone. What do those persistent repurposed Wolffian ducts ultimately become?
They transform into the entire internal male reproductive plumbing. The efferent ductules, the epididymis, the ductus deferens, and the seminal vesicle, as well as the ejaculatory duct. Conversely, the paramesonephric ducts almost completely regress due to AMH, leaving only minor, non-functional vestiges, like the prostatic utricle and the appendix testis.
Now let's look at the alternative, ovary development. This is often described as the default path, but that doesn't mean it's passive, does it?
Yeah. Not at all. In the absence of the SRY gene, the cortex of the indifferent gonad persists and differentiates into the ovary. While the lack of SRY is the initial trigger, this path requires its own set of active molecular factors to promote ovarian development and actively suppress the lingering male pathway. It's a stabilization process.
What are the key molecular factors stabilizing the female pathway?
Factors like WNT4, FOXL2, and DAX1 are essential. WNT4, for example, is critical for promoting ovarian development. It not only suppresses testicular differentiation, but may also directly antagonize the action of the SRY-related pathways. The molecular balance is incredibly delicate.
And how does this default pathway affect the duct systems in the female embryo?
Since there is no SRY, there are no Sertoli cells, and therefore no AMH is produced. Without that chemical bomb, the paramesonephric ducts persist and develop freely.
And the Wolffian ducts?
Simultaneously, since there is no significant fetal testosterone surge, only low levels of fetal and maternal estrogen, the mesonephric, or Wolffian ducts regress due to the simple lack of hormonal support, leaving only vestiges like the apophoron, periphoron, and Gartner's duct.
So the persistent paramesonephric ducts become the female internal reproductive organs.
They do. The unfused cranial portions remain as the uterine tubes, or fallopian tubes. Where the ducts grew caudally and fused at the midline, they form the uterus in the upper third of the vagina. And this fusion process is key, as any failure here leads to many of the structural uterine anomalies we will discuss later.
Let's quickly contrast the fate of the PGCs in the developing ovary, leading to oogenesis versus the male pathway.
The PGCs in the ovary become oogonia, and critically, they enter meiosis prenatally, but then they arrest as primary oocytes. They become surrounded by somatic cells to form primary ovarian follicles. The key point is that this follicle formation is completed prenatally, resulting in a finite, fixed number of oocytes at birth.
And in males.
Contrast this with the male system, where spermatogenesis doesn't even begin until puberty, and sperm production is continuous throughout life. The female starts with all the gametes she will ever have.
Now let's discuss the physical movement of the gonads, known as descent. This happens in both sexes, though dramatically differently.
The descent of both the testes and the ovary is regulated primarily by a cord of mesenchyme called the gubernaculum, which connects the caudal pole of the gonad down to the inguinal region. You can think of it as a biological tether or a guide wire.
The descent is much more dramatic in males. The testes descent, moving from the abdominal cavity all the way to the scrotum.
Testicular descent occurs in two distinct phases. The first is the transabdominal phase, beginning around weeks 12-14. This phase involves active processes, including the shortening and thickening of the gubernaculum and the regression of the suspensory ligament. The testes moves towards the internal inguinal ring.
And the subsequent movement through the abdominal wall into the scrotum.
That's the inguinoscrotal phase. Between weeks 25 and 28, the testis passes through the inguinal canal, guided by the gubernaculum. As it descends, it pushes layers of the abdominal wall and peritoneum before it, which become the coverings of the spermatic cord and the pestes, including the tunica vaginalis and various fascial layers.
And this sometimes fails.
Right. The sources note that cryptorchidism, an undescended testis, is a common observation at birth. But critically, there is a high rate of spontaneous resolution in the first 6 to 12 months postnatally.
And for the ovaries, the descent is far less pronounced.
That's right. The ovaries descend only slightly, halting at the pelvic brim, where they are eventually anchored. In females, the gubernaculum persists but differentiates into the two stabilizing ligaments of the internal female anatomy, the round ligament of the uterus and the ovarian ligament. It ceases its active migratory role much sooner.
Just like the internal ducts, external genitalia also passes through an indifferent stage, where the structures are identical in both sexes before hormonal signals drive the final, highly visible differentiation.
Exactly. And the external genitalia remain identical until stage 23, which is roughly week 9 of development.
What are the three key identical structures present until then?
The three shared structures are, first, the central projection, the genital tubercle anteriorly. Second, the two thin folds, the urethral folds on either side of the urogenital orifice. And third, the paired larger swellings, the genital swellings, which lie lateral to those folds.
The critical turning point for male external differentiation is the presence of androgens, But not just testosterone, it requires a specific, highly potent metabolite.
Absolutely. Male external development is entirely dependent on dihydrotestosterone, or DHT. Testosterone is converted into this much more potent form by the 5-alpha reductase enzyme in the target tissues. This hormone drives the sequence of masculinization. Without DHT, external masculinization fails, even if testosterone levels are adequate, as we'll see in our clinical section.
What is the physical sequence of change driven by DHT in the male embryo?
First, the genital tubercle enlarges rapidly to form the phallus, which will be the penis. Second, and critically, the urethral folds must fuse along the midline to close the urethral groove, creating the tubular penile urethra. This fusion process starts proximally and extends distally, but that fusion is key.
And third.
Third, the genital swellings fuse at the midline to form the scrotum.
And failure of that urethral fold fusion leads to one of the most common birth defects in males, doesn't it?
It does. Failure of fusion, often incomplete, results in hypospadias, where the urethral opening is located somewhere on the ventral shaft of the penis rather than at the tip. This is a direct consequence of that androgen-driven fusion process failing.
Now, contrasting that intense, hormone-dependent process, female genitalia differentiation is defined by the absence of those high androgen levels.
Correct. Female external development is the default passive pathway. It does not require specific high-level hormonal input beyond the low levels of maternal estrogen present. The structures develop with minimal fusion.
Walk us through the female sequence of differentiation.
The genital tubercle enlarges only slightly, forming the clitoris. The urethral folds, instead of fusing, remain separate and form the labia minora. The genital swellings, which are lateral, enlarge to form the labia majora. The urogenital groove remains open, forming the vestibule and the vaginal opening. The key morphological difference from the male outcome is the maintenance of separate, distinct structures.
Finally, let's cover the development of the accessory glands, specifically the prostate and the vagina.
The prostate gland develops in males under the influence of DHT, arising from endodermal outgrowths of the urogenital sinus, starting around 8 to 10 weeks. These outgrowths penetrate the surrounding mesenchyme, which then differentiates into the smooth muscle and connective tissue of the gland.
And the vagina, another complex structure with a dual origin from both the Mullerian ducts and the urogenital sinus.
The vagina is formed by the fusion of the paramesonephric ducts, which contribute the upper 2/3, and the endoderm of the urogenital sinus, which contributes the lower third. The terminal ends of the fused paramesonephric ducts interact with the urogenital sinus, causing the proliferation of cells to form the solid vaginal plate.
A solid plate.
A solid plate, which then undergoes canalization hollowing out to form the final vaginal lumen. It's a process that continues late into the fetal period, emphasizing that even seemingly simple canals are built through complex cellular transformation.
Now we connect these intricate steps to real-world outcomes. When a precise timing, signaling, or fusion steps fail, we see significant clinical anomalies that impact health and reproduction. Let's start with uterovaginal anomalies, which stem from issues with the paramesonephric ducts fusing in the midline.
These anomalies arise from two main types of failure: partial or total failure of the paired paramesonephric ducts to fuse, or failure of the septum that forms between them, the uterovaginal septum, to resorb or break down once fusion is complete.
What does the spectrum of failure look like, and how does it impact function?
Well, it ranges from profound separation to incomplete internal clearance. Uterus didelphys is the most extreme example of fusion failure, where the ducts failed to merge at all, resulting in a double uterus, cervix, and sometimes a double vagina.
And less extreme versions.
Bicornuate uterus occurs when there is only partial failure of the cranial portions to fuse, creating a uterus with two distinct horns. Conversely, a septate uterus occurs when fusion happens correctly, but the central septum fails to fully resorb, creating a dividing wall inside the uterus. These anomalies are very relevant to reproductive outcomes, as they can cause difficulties with implantation, recurrent miscarriage, or preterm labor.
Moving back to the urinary system, what about developmental anomalies involving the ureter and bladder?
A critical anomaly is ureteric ectopia. This is a rare condition where the ureter opens into an abnormal site, such as the vagina, urethra, or prostate, instead of into the bladder trigone.
And that's caused by...
It's caused by the ureter separating abnormally or too early from the mesonephric duct during that crucial absorption phase. Since the ureter doesn't insert correctly into the muscular bladder wall, it often lacks the proper valve mechanism and results in continuous leakage or incontinence.
And in terms of detection, how are serious anomalies of the lower urinary tract identified prenatally?
Fetal bladder and kidney development are routinely monitored via ultrasound. A serious anomaly indicated by imaging is posterior urethral obstruction, PUBO, which is indicated by a thick-walled, dilated bladder and high-resistance outflow. This is often caused by congenital obstructive posterior urethral valves, which essentially block the exit of the bladder.
That blockade must have severe consequences upstream.
It does. If the bladder cannot empty, the pressure backs up through the ureters, causing bilateral hydronephrosis, severe swelling of the kidneys. If left untreated, this high pressure can permanently damage the developing nephrons and collecting system, leading to chronic kidney disease or failure even before birth. It shows the catastrophic fragility of the system when simple plumbing fails.
This all leads us to the complex topic of disorders of sex development, or DSD. The sources emphasize the importance of using this specific terminology now.
DSD is the currently accepted and preferred terminology. It encompasses a diverse group of congenital conditions involving chromosomal, gonadal, or anatomical inconsistencies in sexual development. The importance of the shift in terminology is to move away from older, often confusing and stigmatizing terms, and instead focus clearly on the underlying genetic and anatomical mechanisms causing the variation.
Let's look at the underlying causes. How does the molecular switch failure manifest in the two genetic sexes?
In XY-DSD, where the individual has a Y chromosome and tests but variable external development, the cause often involves problems with androgen signaling. This could be androgen insensitivity syndrome, AIS, where the cells cannot respond to testosterone, or specific enzyme deficiencies that block testosterone's conversion or action.
And in XX-DSD, the internal path is female, with a uterus and tubes, but the external path is masculinized.
Yes, the cause is typically excessive androgen exposure in utero. Most commonly, this is due to the congenital adrenal hyperplasia, CAH, we discussed earlier, leading to virilization of the external genitalia because of that androgen excess from the fetal adrenal gland.
Let's focus on one striking example from the source material, 5-alpha reductase deficiency. It perfectly illustrates how hormonal potency dictates external morphology, giving us a clear narrative of the separation between testosterone and DHT.
This is perhaps the most illustrative case in all of embryology. Remember, the 5-reductase enzyme is responsible for converting the less potent testosterone into the highly potent dihydrotestosterone, DHT, which drives male external development, penis enlargement, urethral fold fusion.
Right.
In individuals lacking this functional enzyme, testosterone is produced by the testes and AMH is produced. So the internal ducts are male, the Wolffian structures persist.
But the external genitalia cannot masculinize fully.
Precisely. Because they cannot produce enough DHT in the target external tissues, the external genitalia fail to masculinize significantly during the fetal period. They may present with female or ambiguous external genitalia at birth, meaning they might be socially raised as girls.
But this individual still has testes producing testosterone. What happens when they hit puberty?
This is the dramatic part. At puberty, the testes start producing massive amounts of testosterone. While testosterone is less effective than DHT in the fetus, high pubertal levels can cause significant delayed masculinization, voice deepening, muscular development, scrotal growth, in some tissues that can respond to T.
So it creates a mismatch.
A profound mismatch between the sex assigned at birth, the internal anatomy, and the dramatic change in external appearance at adolescence. It powerfully demonstrates that while testosterone maintains the internal ducts, DHT is absolutely necessary for early external differentiation.
Finally, let's tie developmental failure to later disease pathology, specifically testicular cancer.
Our sources note a significant clinical link between cryptorchidism, or undescended tests, and a substantially increased risk for developing testicular germ cell carcinoma in situ, CIS, which is the precursor to most testicular cancers.
So where the testes end up matters.
It matters a great deal. If the testes remain undescended or aberrant in morphology due to developmental failure, meaning they never left the abdomen or inguinal canal, the risk of later malignancy is significantly elevated. This reinforces how the early physical environment and cellular history, dictated by embryology, directly influence lifelong health risks and disease propensity.
Here's where it gets really interesting. We've tracked the urinary and reproductive tracts from the very first mesodermal clumps through a dizzying sequence of 3 successive kidneys, pronephros, mesonephros, metanephros, and finally through a hormonal switch dictated by SRY, AMH, and androgens that determines the fate of that initial dual duct system. We saw ultimate biological reuse,
Huh.
temporary urinary ducts becoming permanent reproductive plumbing. It's a magnificent architectural feat defined by razor-thin margins.
So what does this all mean? The complexity reminds us that even minor deviations in timing or signaling, whether it's the lack of WNT signaling needed to maintain nephron progenitors, a failure in AMH production leading to persistent Müllerian ducts, or a deficiency in 5-alpha reductase altering external morphology, can create significant anatomical and functional variations. This illustrates the fundamental fragility and complexity inherent in human embryological development.
Our key takeaways for you should be clear. First, remember the dual reciprocal origin of the permanent kidney, the metanephros. It requires the ureteric bud and the metanephric mesenchyme to induce each other perfectly.
Absolutely.
Second, recognize the temporary nature of the mesonephros, but its vital role as a reservoir for the ducts that will be recycled into the internal male reproductive plumbing. And third, the profound impact of the SRY gene and androgens, which provide the active signal necessary for male duct persistence and external morphology. Female development, while active at the molecular level, is the result of suppressing the male signals.
And this brings us back to where we started, the sheer talent of studying these events. The source material notes that knowledge of human development relies heavily on experimental systems like stem cell-derived organoids due to the sheer difficulty of observing these early transformations in human embryos directly. Consider how much of our basic foundational understanding of this incredibly complex human biology is built on these extrapolated models.
Right.
We know the major steps, but what unknown, nuanced developmental steps, perhaps tiny localized interactions between cells or short bursts of signaling, might still be occurring in the fetal environment that we cannot yet replicate or observe in a petri dish? It serves as a potent reminder that the most fundamental processes of human life occurring in those first few weeks still hold many secrets that future research must uncover.